Hello, and a very warm welcome to today's Cell and Gene Therapy Insights panel discussion, titled Understanding and Leveraging Cord Blood Bank Capabilities and Practices in Cell Therapy Development. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today are a panel of experts who, among other things, will explore the evolving role of cord blood banks in supporting cell therapy development by highlighting their capabilities, practices, and the potential of cord blood as a valuable source for manufacturing therapies. As always, do feel free to pose your questions to our panel using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session today. If you would like to direct a question to a specific panelist, please try to indicate that in your message. And so now I'd like to introduce our panelists. Joy Aho is the Director of Product Management and Strategy at NMDP Biotherapies, with a PhD in molecular cellular developmental biology and genetics and over 13 years of experience in cell biology product development. Luis Hernandez of Cellular Therapies at LifeSelf oversees cord blood connections and therapy initiatives with over 16 years of experience and key contributions to LifeSelf's FDA licensure for cord blood manufacturing. Amanda Connerty, Executive Director at Altiva Biotherapeutics, has over 20 years of experience in cell therapy development and manufacturing, leading process and supply chain operations for natural killer cell-based therapies. Sarah Shields, Associate Director at CCBC and Director of Operations at Cleveland Cell Therapy Incubator, has nearly 20 years of experience in clinical healthcare and research, specializing in cord blood operations and process optimization. So without any further ado, I'll hand over to Joy to kick us off with a short presentation before our panel discussion today.
Hello, everyone. I'm excited for this opportunity to be able to speak with you today on behalf of NMDP Biotherapies. And so I'm just going to do a quick intro to our panel discussion to talk a little bit about cord blood unit sourcing through NMDP Biotherapies and specifically the Cord Blood Bank Alliance. So to start, just wanted an introduction in case you aren't familiar with our organization. The NMDP has been around for over 35 years, really as a global leader in cell therapy. You can see on this slide gives you an idea of just the sheer volume of experience that as an organization that we have in facilitating cell therapies, primarily through cell transplant and now through a fully owned nonprofit subsidiary that I am part of at NMDP Biotherapies. We are taking this infrastructure to cell and gene therapy developers. And part of that, specific to our discussion today, is really related to cord blood and managing a very large registry of cord blood units with close to 270,000 units within that registry. And so as part of that infrastructure and bringing that to developers, some of the services that we are providing are shown here. For allogeneic cell therapy development, really in the cord blood unit sourcing is what we'll be discussing today. But in addition to that, we do work with our large registry to support providing adult donor cell sourcing as well. For both of those, we've got our bioinformatics consulting. We have a wide variety of data available based on that volume that we have experienced through facilitating transplant and now emerging cell therapies over the past several years. And we can leverage that to help define a variety of parameters for developers. And then on the autologous side, we can help facilitate autologous supply chain management as well as collection network qualification, onboarding, and management. And then across both of those modalities, we have CRO services available. So for today, if we focus on cord blood unit sourcing and some of the capabilities that we have, there's really in three main buckets in terms of the types of units that we're able to provide. For cryopreserved, both non-clinical grade units that are units that do not meet FDA eligibility and safety requirements for use in humans, but are very valuable for use in validating and qualifying procedures both research as well as process development. In the middle, you can see the clinical grade cryopreserved units. These are units that are going to meet eligibility requirements for use in humans. They're fully characterized, and they can be for use both in direct transplant as well as for further manufacturing. And then finally, fresh cord blood units, which may meet FDA eligibility and safety requirements and can be used either for use in research or for further manufacturing. And the way that we are able to provide these units is really through the Cord Blood Bank Alliance and the partnerships that we've developed there. You can see here on this slide the 9 different cord blood banks throughout the United States that are part of this alliance and have a shared mission to be able to provide units, this super valuable resource, to developers for further manufacturing and to be able to move the cell therapy field forward. Some of the advantages to the alliance are shown here. So it does provide a single point of access across banks that are part of that alliance. So you get broad access to a much larger inventory than if you were to have to work with individual banks and need to develop those relationships with each bank. This can also then streamline your qualification and audit activities, both for the banks as well as for developers, so that we can do a single qualification through NMDP Biotherapies and be able to leverage that across developers to be able to streamline that for the banks themselves, as well as for developers being able to leverage that qualification that we do and be able to access those units across those banks. We are also able to utilize our supply chain and logistics coordination expertise that we have provided over several years and been able to optimize. And then I mentioned earlier that our bioinformatics capabilities, and this also reaches into the cord blood space to be able to use those that data to look at registry attributes. We can look across the registry, we can look at usage, and be able to look at what, based on you as a developer, the requirements that you're looking at and how that might maximize or limit the number of units that might be available. So I wanted to end on providing an example of that. So this shows you across the full, this is not just Alliance banks, but across the full registry of banks that work with the NMDP and all of the units available in that registry. So close to 270,000, and how they distribute by age. So you can look across, up to 25 years. So banking's been going on, and these there have been studies that have demonstrated, the consistency in the cells of very old units to newer units. But if we just look at those parameters, depending on what you're looking for as a developer, but some of the asks might be, if you are developing a commercial therapy and you're not using these for research, in 2015 is when banks first started to implement commercial consent language in their consents. And so that automatically, if you're needing commercial consent language, can reduce the number of units that might be available from any single bank. But one of the other benefits through the Alliance also allows us to look at what are ways that we can still maximize that usage. So developing reconsenting procedures at the banks to be able to look at how can we utilize more of that inventory for this purpose. We can also, the other advantage of having that broad bank access is that in some cases with the newer therapies, so iPS cells, for example, cord blood can be a very valuable resource for that modality. And having triple homozygous HLA units has been an advantage. And so when we look at that, we can look at the inventory and say, what's available from a triple homozygous perspective, and we go from hundreds of thousands of units down to potentially hundreds of units that might be available for that purpose, which having that broad bank access can have more availability for you to be able to leverage those units for your therapy. So with that, I will stop there, and I very much look forward to the panel discussion.
Thank you so much for that great presentation, Joy, and a great start there. Without any further ado, I'll start today's discussion. So I invite everyone to join me on camera, and you all have. Fantastic. So our first question today, to really set the scene, asks each of you to describe your current experience with cord blood as a starting material for further manufacturing. Joy, if you'd like to kick us off first here.
Sounds good. Yes, so my experience in the past, I have utilized cord blood, just given the unique nature of cord blood in reagent development, and being able to test different reagents utilizing that cell product. More recently is working with the cord blood bank partners in the cord blood bank alliance. I've been able to engage with developers to understand what they're looking for out of starting material, how they're thinking about cord blood units and the types of therapies that they've been using, to be able to look at how might we need to evolve, both from an alliance perspective of adding capabilities, as well as working with the banks themselves to learn a lot about their practices and how we can evolve those for this market.
Fantastic. And Lewis, can I come to you here second?
Sure. Yeah. So, with our cord blood bank, historically, we were just focused on our clinical units. But as everybody or most people know, only a small percentage of the units that we collect overall actually meet the strict criteria to fully take it down that path. Started working with different academic research institutions across the country that were searching for this material, and we've been providing to different researchers for quite some time. And I've also, not only with fresh or frozen, but then also just specific primary cells that they might be looking for.
That's great, thank you. And Amanda, coming to you now.
Sure. So if you didn't hear my intro earlier, it was a little bit breaking up. But so I'm currently at Artiva Biotherapeutics, and we utilize cord blood, cryopreserved cord blood, as a starting material for our allogeneic off-the-shelf cell therapy. We have a few trials in the clinic right now. So that's our experience with cord blood.
Great, thank you. And last but not least, Sarah.
Hi, thanks. So aside from our public cord blood bank, the Cleveland Cord Blood Center, we also have a subsidiary, the Cleveland Cell Therapy Incubator, where we use fresh cord blood units to isolate CD34 cells. We're also set up to work with partners to do contract manufacturing. So we bring their SOPs in-house and can use their protocols, and also have access to our cord blood inventory to perform their procedures. So it provides a consistent and reliable source of cord blood to use for those projects.
Great, thanks for starting us off there, guys. We'll move to our second question now. So what do you feel are the biggest advantages of using cord blood as a starting material for cell therapy manufacturing? And Amanda, if you could kick us off here, that would be great.
So for me, like I mentioned, I'm currently making off-the-shelf allogeneic NK cell products utilizing cord blood. We do have some screening that goes into our cord blood selection. And so utilizing frozen cord blood is perfect for that. Because the units have segments, we can network with the member networks with cord blood banks. They can pre-screen the units for us, at the cord blood bank's leisure. So they can send out the little segments or do spot cards to do the typing that we're looking for. And then I can purchase the units and put them in my inventory and just have them when I need them. So what that allows me to do is really have great control over my manufacturing schedule, my manufacturing process. I can build an inventory of starting material and can, make my scheduling out for the next year or two, versus having to screen fresh donors and things, where you don't really know when you're going to get the material. So for us, it really allows me to control that manufacturing schedule and output of my drug product for my clinical trials.
Great, thank you so much for that, Amanda. Does anyone else have anything to add here?
I think, like Amanda said, it is such a well-characterized product. And as new ideas and research comes up, we have those segments available where we can look at different, parts of the cord blood units, which, you can't always do with like fresh products or other cell types. But, we're very forward thinking in the way that we freeze and save our products so that we have that capability going forward.
Yeah, absolutely.
I
Thanks,
think something that I would add,
Go ahead, Joy.
would just be the biological aspects of cord blood as well, knowing they're high in stem cell, hematopoietic stem cell percentage. There are more naive cells, so depending on what you're looking at, they haven't had that environmental exposure that some of the adult donors might have had. So when you're thinking of things like iPS cells, for example, and you want that cell that hasn't had to deal with that and the potential mutation burden that leads to, and even just that mix of cells beyond that can be super advantageous to a variety of different therapies.
Great, thank you so much for adding that, Joy.
Yeah, and I would just maybe add access, right? I mean, this is it's a natural byproduct that is often just ending up in a medical waste. So I think access is also something that I would add as an advantage. Non-invasive.
Great, thank you for that. Those very insightful answers, guys. I think that was great coverage. Moving to our next question now. What capabilities and experiences do cord blood banks have that may be advantageous to cell therapy developers? And Lewis, if we could stay with you here first.
Sure. Yeah. So I mean, most banks understand the regulatory and they're accredited. A lot of these, for instance, our bank is FDA licensed. And so we understand what the challenges are with regulatory or meeting standards. So most banks have a very experienced quality system in place. And so I would say that would be one of the first things. I think the other thing is the infrastructure that we have built. So over the decades, most banks have the infrastructure for recruitment, consenting, processing, long-term storage, but also the testing that's needed that most people are looking for to fully characterize the material. So I would say the biggest thing is just the infrastructure, the already existing infrastructure. And, although we can tap into a lot of these frozen cord blood units, which is fantastic, we also have the ability to collect moving forward. And I think being able to help developers as they need, if they have specific needs, I think we're in a unique place to be able to help figure that out.
Thanks so much for that, Lewis. And I'll open this one up to the rest of the panel as well. Anything else to add here?
I think there's a huge advantage to clients that want to use licensed cord blood units for their projects. It almost gives you a step ahead in the process because, the bank has already been vetted by the FDA. We already know what's in the product, what's the manufacturing process. So there's, so many different things you're trying to figure out and consider when you're making your clinical trials, when you're making your studies. But that's almost one thing that you can check off the list where, if you use a licensed product, it's going to be good, it's going to work well. So that's why we have a lot of clients that come to us looking specifically for licensed products. But also, our unlicensed products are very good too. So like if you want to use those for engineering runs, that's another option as well. They're manufactured the same way, but they don't have that stamp of licensure, but they're still very comparable products. So there are a lot of options related to that too.
Yeah, I'll chime in on that. I'm currently using licensed units for all my clinical trials, but we do plan to expand that to unlicensed units for the reasons you just described. They're tested the same, they're manufactured the same. And because, I'm interested in the NK cell population and not necessarily the CD34 population, the licensure doesn't really impact me from that. But it was great, when we were first filing our INDs to have that stamp, of a licensed cord to file as our starting material.
Yeah, and I think the other thing too is right, and I think Joy mentioned it, but most banks are doing stability studies on an annual basis. That includes a lot of the IND units, unlicensed units, and we also have decades of outcomes data that help support the quality of those units.
Great, fantastic. Thanks for that, guys. And moving on to our next question, and this one is for you, Sarah. Can you walk us through the current banking process flow? How does this differ across banks?
Sure. So this is something that has changed a lot over time as the industry has changed. I think a lot of banks and ours as well. When we first started, we were just banking everything that came through the door. We just really wanted to build up our inventory. The TNC, CD34, it didn't really matter. But as we've seen, units that are being selected for clinical transplant are the very large TNC, CD34 units. So now we really target those very large units for our clinical bank. We also look at the ethnicity of the units. It's really important to us to have a very diverse inventory. So we're trying to bank as many ethnically diverse units as we can. So high TNC, high CD34, but it's really only the top 10% of the units that we are banking for clinical banking, which means all those other units that are collected are available for research. If the mom consented to research, we want to be able to use those units. We don't want them to go in the trash. We want to be able to use them as much as possible. So that means there's a huge field of units that are available, both fresh and frozen. And I think, Louise, your center is probably the same, small amount going to clinical banking and large amount for research.
Yeah, that's absolutely true.
Great, thanks for that overview, Sarah. That was great. Does anyone else want to weigh in on the banking process flow?
I think one of the things that was, not being a cord blood banker at a bank, but as I've been working with banks and had, been able to talk through and working with developers, is just understanding like all of the coordination and pieces that go into banking. And they're working with, many of the banks are working with multiple collection sites that are our diverse geography across, the United States or, if it's an international bank. And just that coordination and all of the testing and working with the mothers. And there's a lot of different pieces that go into banking that unit that I think are not always factored in when developers are thinking about what they might want out of that unit or that process from a timing perspective or a testing perspective of the mothers or, some of those factors to consider that, that fresh unit is going from point A that might be, several hours away or maybe even farther and being shipped to a central location for processing and testing and all of those various aspects I think are super important and also speak to the experience of the banks and the infrastructure that you guys have built.
And I would add, right, that it's not easy to build that. And so manufacturers leveraging that from the banks, I think makes a lot of sense. Oftentimes I hear that we're a logistics business because of all the complexities.
Yeah, absolutely. I think that really hits the nail on the head there, Lewis. Okay, fantastic. Our next question is around the most common misconceptions about how a cord blood unit is banked. And Louis, can we start with you here?
Yeah, I guess I was struggling with this question a little bit, thinking about it. So, often I tend to think about, the misconceptions are typically, I find more from those that are donating or trying to donate their units. You still hear folks wondering, are you going to clone my baby? Would be one. I think it goes back to what Joy just said. Understanding kind of the complexities of what is all involved with what we do. is probably something that most people don't quite realize. And I think also the strict, again, going back to the regulations and the standards that everybody follows, I mean, they're pretty strict, right? And so we take confidentiality very seriously, traceability very seriously. And I would say that would be one of the biggest or lack of understanding that I encounter.
Yeah, and I'll just add to that. I think there's still misconceptions about private versus public cord blood banks. You know, public cord blood banks were not for profit. There's no fees to the donating mom. You know, we just want to find a use for those units. People that donate, that's what they want to. They don't want it to sit in inventory for years, and they don't want it to go in the garbage. And that's our mission too, is try to find a use for it, whether it's clinical banking or it's for research. You know, either way, you can help save a life.
Thank you, guys. Moving on to our next question now. What challenges surround the modification of current banking practices? What are the biggest pieces that can impact the feasibility of adopting a developer request for modifications? And Joy, we'd like to come to you here first.
Yeah, so some of the things I think that we've run into in working with developers is, I think Luis spoke to it in terms of like the processes that they put in place to support a BLA for hematopoietic stem cell transplant, and even if it's a unlicensed bank, having very clear GMP processes in place for manufacturing of those units. That when you're doing that, and I'm sure developers can speak to this as well, you have to really follow those parameters as you're banking in terms of that. So if you are deviating, that's a bigger challenge, especially if you want to deviate that process for like a one-time thing. But you still want it, not that you couldn't do it, but when it's a really tiny volume, that can be a challenge to be able to support it in a I guess a financially feasible way. I think that's one of the major ones that we've run up against, is how tricky that can be. I think another one, and I know there's capabilities up to a point in terms of testing that unit and wanting that pre-testing. There are some limitations to the samples that are available and what tests you can perform on them. And so just to have, keeping that piece in mind, thinking like you can get, a really high volume of sample to maybe do a sample manufacturing run or something like that. Like that might be something, if you're looking to use a cryopreserved unit, would be more of a challenge.
Thanks for that, Joy. And I'll open this one up to the rest of the panel as well. Anything to add here on the challenges surrounding modification?
Yeah, I mean, Joy nailed it. The onesies and twosies requests. I think we have for most of the staff that's handling this, right? You have procedures, and everybody's trained to follow those procedures very strictly, right? And so whenever you're getting these one-offs, making sure that they're done correctly, but also financially feasible. In terms of what the ask for is probably the biggest challenge we run into.
And I think it's really important that the client knows what they want and exactly all those specifications going forward, because this will happen to us a lot where it's like, okay, we have the TNC nailed down, the CD34, the age of the sample, everything's signed and done. You go forward and now it's, okay, we also need HLA match units. Well, that's very challenging at this point now, because we had this subset identified. You're changing the requirements, so it's going to take some time to work with the client and go back and make those modifications. It can be done, but it just adds more time to the project.
And I'd like to add, so it is challenging, and sometimes there are times where you have to. It might not fully work out, but I would say that we do try really hard to make these to accommodate. And we are flexible And I think, Sarah, I think your center is as well. But it can be challenging.
Absolutely. Thanks, guys. Those brilliant answers. We've got time for some audience questions now. Just a reminder to our audience to keep sending in your questions that you might have for our speakers. We'll get to anything that we can today and answer any other questions via email. And if you'd like to direct your question to a specific speaker, please feel free to indicate that in your message. So we've got our first audience question here. This isn't directed to anyone specific, so feel free to chime in if you've got any thoughts on this question. Any experiences on cord blood-derived gamma delta T cells apart from CBNK cells?
I can talk about that a little bit. It's Amanda. Obviously I'm not I'm not manufacturing gamma delta T cells, but honestly, with cord blood as a starting material, you can choose your favorite cell selection depletion device, and honestly, you can pretty much get whatever cells you want. I mean, for me, I'm doing a CD3 depletion to start my process, but you could certainly do a positive selection for gamma delta T cells. You could deplete out everything and end up with those if you don't want them labeled. But they're really, it would be up to you to just develop your process around which cells you would like to manufacture. And you can do that with any cell on a cord blood unit, really.
Brilliant. Thanks for taking that on, Amanda. Next audience question here. Why NK cells and not T cells for CAR T production? Can we use the T cells in CB units?
I can probably answer that one too. You can. For me, I don't do CAR Ts. I have NKs and CAR NKs. It's just sort of a company decision. We're team NK. There are certainly those who are team T cell, and that's great as well. Can you use these for sort of an allogeneic, CAR T? Sure, you could. You absolutely could. Again, you would have to develop the manufacturing process around that. But it would probably be very similar to what you would get from using any apheresis product. You just have to tweak it for cord blood and modify your cell selection or depletion process. But you could certainly use T cells from a cord blood for a CAR T.
Thanks for that, Amanda. And another audience member is wondering, how about MSC on the blood in the CD34 negative fraction? Could we use the CD34 negative fractions for getting products to cell therapy as well? Anyone want to weigh in here?
I guess I can take that one as well. Sure. So I'm not using a CD34 negative fraction. I'm using a CD3 negative fraction, but you could utilize whatever negative fraction you'd like. I think, like I said, it's a choose your own adventure, honestly, with the frozen material. You could select or deplete whatever cell population you want. And if you'd like to use the negative fraction for your process by depleting out the CD34s, you can certainly do that. It's very similar to what I'm currently doing with depleting out the T cells for my process. And then you would just tailor your manufacturing to facilitate the growth and expansion of your cell of interest. And you can do that in a number of ways. So I don't see any reason why you couldn't use a 34 negative fraction if you're interested in other cell populations.
For MSCs as well, I know, in addition to potentially finding those in the blood, there have been groups that have leveraged the cord tissue to be able to isolate those cells. And that's, depending on the bank, there are banks that are able to provide that cord tissue as well to allow you to isolate the MSCs for further manufacturing.
And we do get requests for the negative CD34 fraction as well. People use it as a substitute for some of their isolated CD34 products that they're testing. So instead of testing that, they're testing a negative CD34 fraction, whether it's for sterility or something else.
Great. Thanks so much for chiming in there, guys. It's great to hear the different perspectives on that. Now we've got an audience question specifically for you, Sarah. How are the units checked for their quality? And how has this changed over the years?
That's a great question. So there's a lot of different potency assays that we do. We do the CFU assay, TNC, CD34 count, viability by flow cytometry. There's a full questionnaire that the mom goes through. So there's product characteristics. Also, the donor eligibility is completed. All of this has to meet our acceptance criteria for it to become available for listing with the NMDP for clinical transplant. So there's a big process that we go through, just checking those samples. Also, if a unit is requested for transplant, there's additional release testing that is performed to make sure it survived the freezing process, repeating that potency assay, performing CD34 counts on the segments to make sure everything is okay and acceptable before it's shipped.
Great, thanks for taking that one, Sarah. Now, next question. Do CB banks have extramural research grants available for supporting technical innovation research with their products?
Yeah, we do look for grants. It is hard to find. I mean, you almost have to put a lot of money in first before you can even apply for those grants to try to get more research funding coming in. So it is challenging. We are able to get some funding through the government for banking of our clinical units. So there are other outlets available through that.
And for us, we have worked with different researchers, and based off of grant applications that they are submitting for, we sometimes help with that as being a support for the grants that they're applying. But direct grants from us, it's rare.
Thanks for clearing that one up, guys. Next question. How can a cord blood bank be processing and banking units that are most able to be used for developing technologies outside of transplant or current specification? Are additional testing segments available? And what else?
So we do store residual samples, plasma, blood spots. There's the segments that are attached to the unit. So those are available, readily available for additional testing. This field is very in tune with what's happening, the research that's going on. So, recently a lot of study info came about natural killer cells. So that's something, how can we bank fresher units? We're very in tune with what's happening, and we try to make strategic plans to meet those needs of the demand of the research that's coming through.
Thanks for taking that one, Sarah. And now one specifically for Amanda. From a developer's perspective, what have you learned that you didn't know when you started? What advice would you give to other developers working with CBBs?
Yeah, I think the biggest thing is being flexible, using the cord blood as a starting material for your manufacturing process. And a few examples, I'll throw Miltenyi out there because I do use their system. There are other systems, for selection, but their devices weren't necessarily designed for a cord blood unit. Even a processed one with red cell removal, it's a very small volume and things like that. So you have to work with the manufacturer of those devices to come up with maybe a custom program to facilitate what you're trying to do with the cord blood unit. The other things, cell washing, cell harvesting, any processes like that, you really have to come up with your own sort of programs for using these different devices that are out there in the market. A lot of things were tailored for apheresis products or for larger volume culture. And I think too, just training your staff on what's the best way to, thaw and wash the cord prior to the start of your manufacturing while maintaining the viability, maintaining, your cell recovery and things like that. And, none of that was something my team was not able to overcome, or that we saw, oh my goodness, this is taking us 6 months to develop. It was just something that we had to do differently from maybe what we had done before with other starting materials. And so I really think it's just, taking a step back, looking at what you're trying to do, being able, being flexible, working with the vendors of, your ancillary equipment that, it's a little bit out of your control, but all of these vendors have been really willing to work with us and come up with solutions. Obviously they would like us to use their equipment, so that helps. But, it's been honestly a really great experience for us coming up, and my team's very excited that they're getting to do something different. And so I think, that's my best advice is just, especially, the handling of the material may be something you have to work on, but also coming up with a manufacturing process. This is, these units are small. And so just really being, focused on how can you optimize using this as a material versus using something that maybe comes with far more cells as a starting material. But like, Joy and some others mentioned, there are some benefits to cord blood. Again, different teams out there of apheresis versus cord blood for a variety of indications and processes. But like she mentioned, these are naive cells. And so depending on what you're trying to do, it could really impact, the outcome of your drug product. So hopefully that answered your question.
That answered it brilliantly, I think. Thanks, Amanda. And now one for Joy. You referred to the commercial aspect integrated to the consent. We understand it has to be broad. Can you describe how it is formulated or even share the document?
Yeah, so it's the exact same language necessarily across all banks. There was some work that was done in partnership with the NMDP to provide sort of an example language of what might be useful to include in the consent. And then each bank has been able to take that and put in place, through their regulatory and legal teams, what they feel comfortable with including in their consent. I think from what I've worked with, the banks and the consents that I've seen, they are relatively broad around commercial use of that product. I think depending on your needs, I would encourage you could reach out to us if you're looking for more specifics around that consent language and if it would meet your needs.
Great, thank you for that, Joy. Now, next audience question. Is frozen cord blood depleted of red blood cells?
I would say the great majority that are stored are either depleted or volume reduced.
Great, nice quick answer there. Thank you for that, Lewis. Next question. Have any limitations been identified specific to frozen CBUs versus fresh?
I think maybe I can start here, and I'm sure Luis and Sarah, you probably have additional thoughts. But, in terms of, I think if we think about fresh versus frozen, one of the things that I had mentioned on, maybe a misconception is that, these units are collected not necessarily right on site at where the processing is happening. And so there are timeline implications when you're thinking about fresh, where it has to go from where it's collected back to where it's going to be, processed and tested, and then to the developer. So, and when you use fresh units, you want to be able to, utilize that product as quickly as possible. So that can be one, potential challenge when utilizing a fresh versus a cryopreserved product. Obviously, with fresh, you have, more access to, if you want to split it in a certain way, or you just want to process it differently than how it might have been processed for cryopreservation, that can be definitely an advantage to fresh over cryopreserved.
Yeah, and I would add the overall recovery. I mean, if you're working with a fresh product and you're looking for specific cells, you're going to be able to recover a lot more and retain a lot more of those cells versus if you're working with a frozen. But to Joy's point, the logistical aspects of all this, frozen makes a lot of sense for a lot of reasons.
And I think it's just important to know your cell type and the stability of the cell that you're looking at. For example, the CD34 cells are very stable. So, I mean, you could be isolating them a couple days out versus other cells that you really want to isolate within the first 24 hours or so. So it's important to know what you're looking for in the stability.
Brilliant. Thank you very much for those answers, guys. And next audience question. Legally, who owns the CB units in the public CB banks? As they're not used for transplantation, can we use them for ATMP development without asking the mother?
I think that goes with the consent. So, our consent, it's a public cord blood bank, and it's very clear that you're giving up ownership of that cord blood unit once donated. So it's definitely covered by our consent.
Agreed.
Thanks
Yeah,
for
and
that,
I
guys.
think, Joy, you showed the timeline. I'm sorry. Earlier of the consenting process, and for me, when I'm requesting units, I do make sure that those have been consented for potential commercial use, because if it is an older unit, but the banks are always great at verifying that for me. But that is a question I ask when working with banks. Like, I'm only interested in this because I don't know, when I'm making master cell banks, how long I'm going to have the material, and so it's just part of the questions you can ask.
And we can go back and reconsent the donor as well if there is, a particular unit someone's interested in. It was collected a long time ago. They didn't have the commercial consent. We can reach out to that donating family and see, yes, do you agree to commercial consent for this unit? Yes or no, and then go from there.
Fantastic. Thanks for adding that there, guys. One for Joy now. Can NMDP provide historical outcomes data to labs who don't receive such feedback from treatment facilities?
Yes. So through a partnership with the Medical College of Wisconsin, the Center for International Blood and Marrow Transplant Research, the CIBMTR, they have collected transplant outcomes for over 30 years. And so they have all of that data available, and they are able to, we partner with them to be able to provide that to developers in different ways. And then we can also leverage the bioinformatics group that we work with, depending on the specific question that you're looking to ask. And then, be able to tailor that. Like if you're, interested in cord blood specifically, they can work with you to develop different, put together different research questions and be able to leverage that data.
Thanks very much for that, Joy. Tons of audience questions coming in here, and rest assured, if we don't get to your question today, we will answer it via email. Next up, is the cryopreservation used in frozen cord blood contain DSMO? If yes, is that a problem for users?
I can answer. So it does contain DMSO. If it's frozen, it's fairly typical, but it has not been an issue for us. Typically, what we do, especially when buying a licensed unit, is we just follow the manufacturer's guidelines for thawing and reconstitution of the cord, and then we assimilate that into our process. We've experimented with washing off the DMSO versus just diluting, based on the instructions from the vendors, but it didn't really impact our process. So no, it hasn't been a problem for us with the DMSO content.
Yeah, I think, I would say it's probably, if it followed properly the washing protocols, it would be a small residual that would be found, right? I don't know if you've tested to see what that residual might be.
We haven't specifically tested, done a residual assay for that. That I recall. We could have done it preclinically a few years ago, but like I said, it hasn't impacted us at all. Like I said, following that dilution, sort of washing process, it diluted for what we're about to do, using our cell selection, it's negligible. So it really hasn't impacted us at all.
Thanks for clarifying that, guys.
I think,
apologies,
sorry, I was just
Joy,
going to say, and something to keep in mind is that these units have been used for direct infusion to transplant patients for 20 plus years, and and through we have that outcomes data and their safety data and all of that I don't believe it has highlighted any issues.
Fantastic. Thanks for clarifying that, everyone. Now, next question. How many CB banks are also banking cord tissue mesenchymal cells?
We are not. So I can speak for our bank. We do have access to the tissue itself, but we're not banking.
We do get quite a few questions about that, but we're not banking it at this time either. They're just, for us, hasn't been enough of a demand for us to move our resources to that area.
Yeah, I think I can confirm across. We do have some of the alliance members that have indicated that they're they have the capability to provide cord tissue, but it is definitely a minority. I guess here I would just also mention that, you know, one of the kind of the goals of having this Cord Blood Bank Alliance as well is looking at industry trends and looking at, you know, to Sarah's point, you know, where is the demand, and then being able to work across banks to be able to look at what should we start working to implement from a capability perspective that we think is going to be really advantageous to the market, so that, you know, we can do that on a broader scale versus one bank really trying to take that on themselves and figure it all out. How can we, you know, partner as a group to be able to do that?
Yeah, and I think, you know, through the alliance here, we would certainly be open to supporting that, right? If it makes sense.
Great, thank you very much for those perspectives. Next question now, and this is a bit of a long one, so bear with me. Do you perform the CFU assay as a qualification assay for assessing the potency of cord blood units? At what point of the collection process do you do this potency testing? And recent updates to guidelines from FACT and JACEIE now require the CFU assay pre and post processing steps. How are cord blood banks and cell therapy developers reflecting this change to the guidelines? Anyone want to chime in here?
That was a very long question. I will say that we do the CFU assay. It's post-process pre-freeze sample. We also do it as part of our release testing on a segment. I don't remember the rest of the question. Louise, help me out.
No, you're good. We do the same. And, you know, we've always been reporting CFU. So I think we were already meeting the standard regardless of the update. But it's a 14-day assay for us. And so, and we have to have those results before being able to get it through final review and listing on the registry.
Mm-hmm.
Thank you very much, guys. And are cord blood banks able to provide units to international developers?
Yes, absolutely. There are some differences, especially in the infectious disease testing requirements between international and domestic shipments. So those are some things that you do need to be mindful of when you're looking to source cord blood units from the US. But yes, we can provide those, and I think a lot of banks can.
Yeah, and I think, and we've got some experience even just from the clinical side, what those differences are and some of the additional documentation that has to be filled out. So, but absolutely, we can support that.
Great, and I think we've got time for just one more audience question here before we wrap up and get your final thoughts. So even with an optimized cryopreservation and banking process, cell loss does occur. Since cell population is homologous, is the cryopreservation and thaw impacting a specific subpopulation of cells? Can anyone speak to that?
I don't know that I can speak on that one. Sarah?
Yeah.
I'll look to Amanda.
Yeah,
there we go.
So it's hard to say, right? Because you have to remember, like, a cord blood came from one individual, and everyone is different. And for me, in my experience, the cord bloods are all different. You know, for licensed units, obviously there's a minimum criteria it has to meet. But as far as the composition of the unit, what I found is they're all different. The CD3 ranges are different. The 34s are different. The NKs are different. T's, B's, mono, again, everything is different. Now, is the result that I get post-thaw a function of the cryopreservation and thaw process, or just a function of the donor differences? You know, I consistently get similar viabilities post-thaw, similar cell recoveries of total cells, because again, the individual populations are different. So I think when you say like the cell population's homogeneous, it depends what you mean by that, because it's homogeneous for that particular individual. But if I'm comparing two, the homogeneity is not the same for each of those. And so, for me, it's a function of my manufacturing process. How do I accommodate the differences? And so, like I said, for us, removing the T cell fraction and just manufacturing with what is remaining has worked. But if you're interested in a specific cell population, like if you want to positively select a cell to use, you're going to find that they're every unit that you, and it's not necessarily it's just a function of, I think, being a human probably to some degree. But again, it hasn't really impacted me in any way. We just accommodate for that. But yeah, I haven't seen any issues related to the freeze thaw. Hopefully that answered your question.
Great, thanks for that, Amanda. And now, to close today's discussion, I'd like to hear what our panel is most excited about for the future of cord blood in cell therapy. Amanda, if we could stay with you here to kick us off.
Amanda, I think you're on mute.
Sorry, how did that happen? Sorry about that. I'll start again. I don't know how I got muted. Yeah, so what I was saying, yeah, I'm very excited for the future of cord blood. Obviously, being a cord blood therapy, cord blood derived therapy developer, for me, just seeing all the interest, seeing the excitement around the potentials of cord blood, it gives me, makes me optimistic that for me, as using cord blood as a donor source, that material is not going to be going away anytime soon. I think, engaging the banks and showing them it's great for transplant, and I don't want to take away from that, but it's also great for so many potential other indications, other cell types, other therapies, CAR T, CAR NK, MSC therapies. It's really, in my mind, limitless. And again, with using frozen cord bloods and just having, like Joy mentioned, hundreds of thousands of units at your disposal to test and optimize your therapy, it's really something you don't see with other cell starting materials, because we just don't have hundreds of thousands of banked other donor material at our disposal, to really come out with such a potentially, life-changing therapy for someone in need. So
Thanks so much for that, Amanda. And Joy, over to you.
Yeah, I would echo a lot of what Amanda said. I'm really excited for growth, and as more developers realize the advantage of using this really valuable resource that there's just so much potential for, and be able to see that grow. I mean, we've seen some really great work coming out of Artiva. Katie Rezvani recently publishing some great clinical, early clinical results from their work at MD Anderson, advancements in iPS cells that are leveraging this. So I think as that science advances, just the capabilities and the ability to leverage such a valuable resource, I think is going to be, just amazing as we move forward.
Thank you for that note, Joy. And Louise?
Yeah, I mean, I think Amanda and Joy covered it well. Just being part of this is exciting. I do think the sky's the limit. And I think, the ability for us to be involved in something that I believe is going to continue to help patients in need, and the population of those patients increasing is just exciting for me to be part of it. So.
Absolutely. And Sarah?
And I'll just add too, I think the collaboration across this field is just tremendous. I mean, between the public cord blood banks, between NMDP, the clients, the manufacturers, it's a very rewarding field to be working in, and just seeing so much, scientific discovery that's coming out of it. It's very inspiring.
Fantastic. A great note to end on. Well, thank you very much, Louis, Amanda, and Sarah and Joy, for a very informative and thought-provoking discussion today. That's unfortunately all we have time for, but any questions that we didn't manage to get to, we will reply via email. Today's webinar will be available on demand tomorrow, so look out for an email from us with the link. All that's left is to thank our speakers once more for a great discussion today, and thank you to the audience for listening. We hope you'll join us again soon.